Hello everyone, welcome to the channel Microbial World. In this video, we will be discussing about capsule staining. Capsule staining is a special staining technique used to visualize bacterial capsules which is located outside the cell wall of certain bacteria. Capsules are often difficult to see using conventional stains because they are made of polysaccharides or polypeptides and do not take up most of the stains. Capsules are important for bacterial virulence. Bacterial virulence refers to the ability of bacteria to cause disease. So capsules play an important role in virulence by helping the bacteria to evade host immune system by preventing phagocytosis. Capsules are composed of polysaccharides. It forms a gelatinous sticky outer layer around certain bacteria. Capsules enhance bacterial resistance to desiccation, which means extreme drying. It provides protection from phagocytosis. Phagocytosis is a process by which certain cells called phagocytes engulf and digest large particles such as bacteria, dead cells, or debris. This is a key part of the immune system and it helps the body to fight off infections and remove unwanted materials. So the capsules are preventing the phagocytosis process. Capsules also contribute to bacterial adherence to surfaces, which refers to the ability of bacteria to attach to surfaces, host cells, or tissues. This is also a very crucial step in infection, that is allowing the bacteria to colonize and resist being washed away by bodily fluids. Capsule staining methods. There are several methods for capsule staining. Negative staining with India ink. This method does not directly stain the capsule but highlights the background, allowing the clear capsule to be visible. The His method combines a simple bacterial stain, for example crystal violet, with a negative stain, for example India ink or nigrosin, to highlight the capsule. The manual staining method uses a combination of acidic and basic stains to highlight the capsule. In this video, we will be discussing more about the India ink method of capsule staining. India ink method is a type of negative staining where the capsule remains colorless or clear and the background is dark due to the use of India ink or nigrosin. The materials required for India ink method includes India ink or nigrosin, negative stain, glass microscope slides, sterile inoculating loop or swab, bacterial culture, liquid culture or growth from an agar plate, distilled water, Bunsen burner, slide rack. Procedure. Take a clean glass microscope slide and place a small drop of India ink or nigrosin on one end of the slide. Mix a small amount of bacterial culture into the ink. If you are using a liquid culture, use an inoculating loop to collect a small amount of bacteria from the culture and mix it into the India ink drop. If you are using an agar plate, take a small amount of bacterial growth using the loop and mix it into the India ink drop. Add distilled water if it is necessary to achieve a smoother suspension. Now take a second slide and place it at a 45-degree angle to the slide with India ink mixture. Now using this angled slide, spread the drop across the slide by pushing it gently across the first slide. This will create a thin, even smear. Allow the smear to air dry naturally in open air. This will ensure that the capsule's integrity is maintained. After the smear is dry, place the slide under microscope. Start with low magnification 10x to locate the area of the smear and then move to oil immersion 100x to examine the cells closely. Look for clear halos, that is capsules around bacterial cells. The bacteria will appear as light colored cells and the capsule appears as a clear halo around the cell. The background will be dark due to India ink stain. Precautions. Do not heat fix the smear. This could cause the capsule to shrink or distort, leading to inaccurate results. Ensure that the smear is thin. This is important for clear visualization of the capsule, as a thick smear can make it difficult to see the capsule. Proper air drying is essential. Rushing the drying process, for example using heat, can lead to distortion of the capsule. Examples of bacterial species with capsule includes Streptococcus pneumoniae, Klebsiella pneumoniae, Haemophilus influenzae, Neisseria meningitidis, Bacillus anthracis. So this is all about capsule staining. Thank you for watching.